Welcome children. We are going to study about the search of the genetic material. Now, through the experiments done by Mendel and Morgan and all, it was established that chromosomes are the genetic material. But the real nature of the genetic material was still unknown. The experiment done by Frederick Griffith in 1928 proved that DNA is the genetic material. So, Griffith's transforming principle is an important experiment in our understanding of the genetic material. Now, he conducted his experiments on a bacteria that is responsible for causing the pneumonia. So, the bacteria he took was Streptococcus pneumoniae. Now, this bacteria was known to cause pneumonia and it had two strains. Yeah, he was capable or he was able to isolate the two strains, and the first strain was the S strain or the smooth strain, and the R strain or the rough strain. The name rough and smooth is based upon the colonies, because the bacteria of smooth strain, the colonies appeared smooth, and this smoothness was due to the capsule which is present in the case of S strain bacteria. The capsule is made up of polysaccharide. So this polysaccharide capsule made the colonies of S strain appear smooth. The R strain did not have any capsule, so no capsule present, and therefore the colonies appeared rough. Further, it was also found that that is the S strain, was capable of causing infection, therefore it was virulent and pathogenic, since it was capable of causing the infection, whereas the R strain was found to be non-virulent and non-pathogenic. That means this strain of bacteria was not capable of causing any sort of infection. Now, for his experiments, he first took the S strain, which was known to cause the infection. So he introduced the S strain into the mice. He took mice for all his experiments. So S strain was introduced into the mice, and since it was virulent and pathogenic due to the infection, the mice died. The result was well expected. Now, further, he introduced the S strain into the mice again, and since it was non-virulent and non-pathogenic, the mice lived. That means there was no infection. In the second set of experiments, he killed the S strain and it was heat killed, and this strain was introduced into the mice. Now, since the heat killed S strain was the S strain was heat killed, the mice did not had any infection and mice lived. In the further experiment, he used the S strain, which was heat killed, and he mixed it with R strain, which was living. That means the live R strain. Now, the expected result of this when introduced into the mice, since the R strain is heat killed and is not capable of causing infection. We have seen over here S strain, which was heat killed, when introduced into the mice did not cause any infection into the mice. But over here again, when he introduced the mixture of heat killed S strain and R strain, it was expected that since S strain, which is heat killed, is not capable of causing any infection, and R strain, although live, is non-virulent and non-pathogenic, will again not cause any infection, the result expected was that the mice will not have any infection and the mice would live. But to his surprise, the mice died due to infection. Now, this means that there is some factor in the S strain that had caused some transformation into the R strain which was living, and as a result the R strain was transformed into virulent type, although it was non-virulent. So he was not able to understand the nature of this transforming material that caused the transformation in the R strain. Although he was capable of, or although he was able to justify that there is some transforming substance that had made the change into the R strain from being non-virulent and non-pathogenic to virulent and pathogenic. So he was not able to give the conclusive biochemical nature of this transforming substance. Later on, MacLeod, McCarthy and Avili, these three scientists performed series of experiments to provide a conclusive nature or the conclusive biochemical nature of this transforming principle. In their experiments, from the heat-killed S strain, which was heat-killed, they isolated the components, that is the RNA, proteins, DNA, carbohydrates, etc. So they isolated all the components from the heat-killed S strain, and to this was added the R strain. These were added to the R strain which was live. Now RNA was added, protein was added, the DNA and the carbohydrate, and further. Now he found that this introduction of RNA into the R strain live did not cause any transformation. The protein further did not cause any transformation. DNA caused the transformation and the carbohydrate did not cause the transformation. But still, to prove the result conclusively that DNA is the transforming principle behind the conversion of non-virulent R strain into the virulent type, they used the enzymes, and they found that RNases, that is the RNA digesting enzyme, the proteases, that is the protein digesting enzyme, that is protease, and their RNAs. These enzymes, when introduced, did not affect transformation. So they did not affect the transformation, whereas the DNAs, that is the DNA digesting enzyme, that affected or that inhibited transcription, that inhibited transformation. So this proved conclusively that DNA is the genetic material and it is present on chromosomes, and therefore the traits are passed from one generation to other generation with the help of DNA. So this is the first set of experiment that conclusively established that DNA is the genetic material. Later on, several experiments by Hershey and Chase and Michelson and Stahl, all that proved conclusively that DNA is the genetic material. Thank you.